Good afternoon, everyone. I am Majdi Al-Abadi from the University of Georgia, and I'm going to be walking with you today through some of the current methods and approaches for sequencing and surveillance and general pathogen sequencing. The most important thing that I will be focusing on is the challenges and the issues with the current methods and what are the potential solutions for those methods in the market. And in the second part of my talk, I will basically talk about what we are doing in our lab in order to address these concerns or these issues with the current techniques and what we are developing. And I'll finish by highlighting some of the methods that we are expect to see in the near or maybe mid-near future, and they should have major effect on the surveillance of pathogens and sequencing of pathogenic genomics in general. The outline of my talk, the first part will be focusing on the sequencing approaches, and I will cover both the untargeted or targeted approaches with general discussion of their workflows, their limitations, and some of the solutions to the major issues with each one of them. And then I will talk about the current pipelines of bioinformatics analysis, the similarity between them and the current suites of analysis that can be utilized for those. And basically, most of people would use some tools that can do everything, but I will be basically highlighting the different steps in these tools. Then I will talk about some of the current application of machine learning in sequencing of pathogens, and then I will move to our own work in the lab. So to get to this, let's talk about the first approach, which is the metagenomics approach. And the concept of this approach is that you have a total nucleic acid from a clinical samples. You have no prior knowledge of what's in these samples, and you just sequence everything in the sample. And the platforms that you can use, all the current method sequencing technology have methods for that or have techniques for that. Illumina, Element, BacBio, NT, they all have kits and reagent and method that you can use to do metagenomic sequencing. It's not a big deal at all. The most important or the most difficult thing about this technique is the sensitivity, because you're trying to sequence everything in the sample. Your target or the pathogen of interest or the pathogen that you're looking for, or might be the cause of the disease or the outbreak, might be of very low in the background, and the sequencing of metagenomics or trying to sequence everything might end up not capturing this rare signature or rare signals of these microbes, especially if your sample is not representative to the peak of the infection or whatnot. So in other words, have very high CT value, then you probably unlikely to capture these viruses. And it requires really a very significant enrichment upfront in order to solve this problem. But as an advantage, you can get a quick idea of what's in the sample. You might be lucky to identify things that you wouldn't expect to see in these samples. And it's probably the quickest and the cheapest methods out there that use for this metagenomics in general. And all platforms have ready-to-go kits, and you don't really need to do any upfront design for anything. You have total RNA or total DNA, you can sequence that very quickly. As I said, the technical challenge of that is how to minimize the host material, how to remove all the unwanted sequencing data and not sequencing all that material that you don't need, which can and will for sure obscure the signal of the target virus or the virus that can be, or the pathogen that can be of interest in this case. The overall workflow for this is you can start either by total RNA or cells, and then you do crude cDNA synthesis, and depending on your sequencing method. If you're doing short reads, you probably will need to do some sort of fragmentation unless your initial material is actually compromised or fragmented. That way you can skip the fragmentation step. But if it's not, you do fragmentation. If you're doing back bio or ONT, you don't need this fragmentation step. And then you enter into the barcoding and amplification and then do the sequencing afterward. It's very straightforward. There is nothing really difficult about this workflow. Uh, as I said, I will talk about really the most difficult part or why this workflow is not really optimal for surveillance or for really discovery. And the main reason is really the high background of the host and ribosomal RNA and things that you might not really want, and they can end up constituting the majority of your samples. And in sequencing, because it's done just probability-wise, the most abundant DNA or nucleic acid will be sequenced the most. So if your target, which will be less comparing to the host, then you're probably not going to get that. So to deal with this problem, you have to basically do some depletion of the host and ribosomal RNA. And there are multiple ways to do that, and we can categorize them as one is the hybridization-based methods, and there is the ribo zero, the ribo minus, and NibNext. Those are all methods that use bioannotated probes to capture or target and capture ribosomal RNA and boil it down, or use RNase H to digest that and remove it from the sample. And that really kind of removes anywhere between 70 to 90% of the ribosomal RNA from your samples. Depends on the samples, depends on the efficiency of the probes that have been designed, and depends on the lab technique as well. Then you can end up removing up to 90 or even more than 90% of the ribosomal RNA from your sample. That really provides some sort of good enrichment in a way. The next approach is very similar to that, but it's actually you capture and then instead of really digesting and removing, you capture that and then use the supernatant of Tarwat or the unwanted sequence, and the supernatant will have your wanted sequences and then use that as your material for the next step. So it's kind of subtraction way of your samples. And there are a couple of kits available for that. One among them is the Sure Select from Agilent. The other one is the enzymatic, and that's the Fast Select from QIAGEN is a representative from that. And this method is either blocks the ribosomal RNA from being synthesized into a cDNA, or use X nuclease to basically digest the ribosomal RNA from the 5' end. And those are very good, very efficient methods, but they are also very expensive kits to buy and work with. I think the QIAGEN kit is like $3,000, $4,000 for about 96 reactions. So it's quite expensive really to work with. The final, and I think one of the very promising methods is use the CRISPR-Cas9 to direct the CRISPR protein to cleave and degrade the ribosomal RNA and the host RNA. So one of the things about this method is that it can enable you to basically target both ribosomal RNA, different types of ribosomal RNA, as well as some of the host. If you have genomic or even messenger RNA from the host in the sample, you can target them. So there are multiple pieces into this design, so you can create a CRISPR clean kit, and there is a couple companies that actually provide those kits ready for now that you can buy and use them, and they have like a combination of CRISPR-Cas9 for the 16S, 28S, 18S, and all the ribosomal suite, as well as some of the mitochondrial and the human DNA as well. So the way this works is basically you start by designing a guide RNA. This guide RNA is complementary to the sequences that you want to remove, and this guide RNA is loaded into the CRISPR-Cas complex, and then you add this to your samples. Some people add it to the final library, and once you make your all final library, you can treat it with the CRISPR-Cas, and in some ways also you can add this before producing your final library at somewhere between the barcoding and the PCR. Once you have a double-stranded molecule in your library process, you can always use that and you add this, and this CRISPR-Cas will end up, targeting all these ribosomal genes and any other molecules that you included in the designs. They will be target and they will be degraded or cleaved, not being suitable for sequencing after that. This is a very efficient method, very good, but it's still really, really expensive method. And if you're gonna do this on regular basis in your lab for surveillance or for whatnot, that probably gonna cost a lot unless you really master this in your lab and be able to create the CRISPR-Cas complex yourself in your lab, which you can do. You design the guide RNA, and then you can order this from companies like IDT and other companies and use this in your lab. But if you're gonna buy like commercial kits, like the one that provided with jump codes and other providers, those are very, very expensive kits, and I don't see them being very visibly used unless you have really tons of money at your disposal. The other one method that I want really to bring to your attention and you highlight is called RiboPub. And this is basically a DIY method. So RiboPub is very similar to the hybridization method, but you can actually design this yourself in your lab, and there is a tool that is in GitHub now that you can use to design for any host that you want, or for any species that you're working with. If you're working with flu, human flu, or avian flu, you can design this for the ribosomal RNA, or even for the human host DNA, or for the avian host DNA, and then design that and synthesize this and have this pay the upfront cost for making these in your lab. But the usage of these is very simple, very easy, very straightforward. And we do use this in our lab for a lot of ribosomal RNA from bacteria. When we do microbiome analysis, we do this, and we basically, I think we paid about $2,000 upfront, but the reagents are available for us all the time. And it's really straightforward. You basically have the ribosomal RNA database from the species that you're working with, and then these ribosomal RNA are kind of clustered and when go through some sort of blocking, and then you design probes on the unique regions of this, and these probes are bioadenylated and ordered, and then you treat your samples with this just like any commercial kit you will buy, and remove all the unwanted or uncaptured ribosomal RNA in the elute, and then you have your basically sort of like 90% enriched samples. And this a figure from the paper that really came up with this approach, and you can see on the bar chart on the upper left side, the 1, 2, 3 input there, you can see the amount of the ribosomal RNA, and after depletion, you can see that other RNA has been really more representative in the samples. And you can see that in the bottom as well, the difference between the input and the depleted RNA. So it's very efficient, and I think very suitable for labs that do a lot of work with metagenomic sequencing, and it's really very effective in, especially in lowering the cost of the depletion. The second is a group of tools or group of approaches are the targeted approaches. And the targeted approaches really differ from the metagenomics is you really need to know, or you have to know what pathogens you're looking for or pathogen you're trying to sequence. And it can target, a group of pathogens based on their variants, or it can target a single virus or a single pathogen. Whatever it is, you can even use it. I think it's mostly suitable for viruses, because if you try to do targeting with pathogenic bacterial genomes or whatnot, those are a quite large genomes, and it will be very costly in this case. But for viruses, I think it's very efficient, and you can use either of these methods, and they can be used with all current available platforms: Illumina, Element Biosciences, ONT, and PacBio. In our lab, we do almost everything now with Oxford Nanopore. just because we have a better control on the sequencing material and the sequencing approach. So we prefer to do that, and we try to optimize our development to meet the requirements of the Oxford Nanopore sequencing. The limitation of this, of course, is you really have to know a little bit about designing either primers or probes or MIPS, and you probably need to, once you design and optimize and test, you need to pay some upfront cost to make this material abundant in your lab, and you can use this regularly. The other thing also is some of these approaches will require you to regularly update the design and then use like in light of reappearance of new variants or new pathogens, you probably need to update your design and make sure that it can capture these variants. The advantage, it's really highly sensitive, and we, as I will show later, we have really good success with things that have CT value of 30 and above to get complete genome capture, and it doesn't require any ribo depletion or any host depletion, so that kind of headache is is out of your way because of this targeted approach. And the only issues that, as I said, is you have to update the designs, and some designs can result in dropouts where if the primers or MIPS or oligos happen to be in a place where there is some variation or some variance in a new strain, then you probably end up having dropouts, which can cause gaps in the assembly and whatnot, that and you would require to regularly update your design in this way. And the workflow is very similar. You first, the green box that I have there is the one that you have really to master at the beginning, how to design and synthesize your oligos and find a cheap way to get your oligos from the current vendors there, and make sure that you have the tools to be able to design those regularly and update them as you needed. But once you have this, the workflow starts with having your RNA or cells, and then you hybridize with your oligos and you capture the targeted, and then you do amplify and barcode, and you're ready for sequencing. These major steps are the same regardless of sequencing technology you're using. You can be using Illumina, Element, ONT, PacBio. It's very much the same everywhere, and that's obviously technologies have slightly different modifications here and there, but these are the major steps that you do if you are using this approach. So let's talk about these targeted approaches. The first one is the PCR-based approach, and there are a number of kits available for that. Among them are the RTEC, the Primal-seq, the Ample-seq, the CleanPlex, and the Normalize panel. And those are basically depend on designing a group of overlapping primer pairs, and these used to amplify overlapping products. And because of the overlapping nature of the amplicons, you can basically assemble them afterwards and creating a complete genome from this assembly. And there are multiple kits available for this that you can use. And I believe that the RTEC was heavily used during the COVID outbreak, I believe so. And I think also they are available for both short reads and long read sequencing. The second approach is the hybridization based, basically. In a way, it's really very similar, but instead of amplifying, you pull down the target from the mix that you have, and once you have the target, you end up amplifying the target again or cloning it and making library and sequencing that. And these are examples for this are the short select, the twist viral panel, and XGen from New England Biolab and MyBates and other kits available for that. So you capture, pull down, and then wash, then release, amplify, and make libraries from that. The final approach is the using the molecular inverted probes. And this, I'm not going to talk much about this because I will be bringing this back later on in the talk. But it's really combines the benefits of both approaches, the BCR and hybridization based, and it is also a little bit more specific, as you can see from the diagram, that there, each probe has two legs there, and that means each target has two specific regions in the genome and the amplification between them. And the backbone really ensures that those targets are hybridized at specific distance from each other. So, as the major challenges with the targeted approaches, as I said, target dropouts that can often results in gaps in your sequencing, and you have to update your design regularly. And as far as I know, the current state of these targets are single species. So you have sure select or whatnot, or Arctic for SARS or for flu. So there is not really anything that can target more than a one species. Probably all of them can target variants of the same species in the same run, but not more one than a species in the same sequencing run. And there are good reasons for that. And also, if you want to go with targeting sequencing, unless you have some development done in your lab, you probably end up counting on a commercial solution provided by those companies. And even if you wanted to do this in your lab, the available resources for you to design overlapped primers and large scale, high multiplex primers, BCR primers, there is not a lot of tools available there. Most will divide or depend on primer 3. And really, for you to use primer 3, you efficiently, if you don't have a commercial platform that like Genius or other that have this application in it, then you would want to use this in the command line, and there is all sorts of issues with that. Other than that, you will just have to buy those kits from public or from commercial vendors. For the MIPS, on the other hand, it's actually a lot worse in terms of the available resources out there. There's only one MIPGen tool that you can use, and it's actually designed to target single genes and not really whole genome sequencing. There is not enough information out there or even commercial solutions for that to do whole genome sequencing using MIPS. Even though it seems like to be the most or the best method out there, but there is not a lot of resources for that. And I will talk about this in a few slides, basically. As for the bioinformatics approaches and how you analyze this data, whether you're working with metagenomics or enriched or targeted sequencing, the approaches are very similar, right, with slight differences from here and there. But if you're talking with metagenomics, you can be working on metagenomics where you're sequencing everything in the samples, or the other light flavor of that's called microbiomics, where you're just targeting the 16S genes of these samples. So either way, you start basically by cleaning your sample, your reads, removing all the bad sequences, the homopolymers, all the adapters, all the things that you added in your design. Everything has to be basically removed up front, and you can use multiple tools for that. And after that, you basically start removing the host material or the ribosomal RNA, assuming you've done some host or ribosomal RNA depletion in the experiment. You still have some left, and you probably need to remove this up front. And then you go next to map your cleaned material or cleaned reads to the reference. And if you're doing metagenomics, then you probably need a database that has reference genomes, and you can use the NCBI, RefSeq, or the GenBank, or the ENCODE, or the GenCode database. Or if you are doing something like taxonomic sequencing, like 16S, there are a few databases that you can use, and among them are the Centrifuge on the Kraken2 and the Meta-Fan-Allen databases. Those are about 16S, and they are like databases and tools at the same time, basically. So you can use those to do the mapping and to the databases as well. So you don't need really to use Botai or BWA or Minimap2. And the difference between Botai, BWA, and Minimap2 is really depends on the type of sequencing you have. If you have long reads, you probably use Minimap2. If you have short reads, you'll use Botai or BWA. And after you do this mapping, you end up having the clustering or assembly. If you're doing a genome assembly, you need metagenomes mags, and you can do assembly. And there are a bunch of tools out there can be used, among them spates and metaspates. If you're doing classification, you basically end up having cluster, and you taxonomic clusters will convert all the sequence data into taxonomic units, and they call them O2Us, and they have different names nowadays. So, but that's the step, and then you go after that to do the variant calling to identify the subtypes or the variants of the species, and then calculate the abundance using statistical packages, and go and basically, sorry, do the abundance and do statistics using to get the alpha diversity, beta diversity, and all this other information that you need for your analysis, assuming that you're really targeting multiple genomes and not only one species of interest. Once you find it, you got what you need there. And there are multiple tools that you can use, and most of them are command line based, and actually also in R. We all have lots of R packages for that. In addition, some available commercial software can help you do this quickly without having to worry about the details of coding, of R coding or whatnot. If you are doing the targeted approach, it's very much the same. You just have to remove a couple of steps there that you are not interested in now, because you don't have host material, you're not doing taxonomic classification, it's all genomics. Then you remove those steps, and everything very much is the same. You do the primer. The most important step in this that make difference from the metagenomics one is, here you have lots of probes or primers in your sequencing. The primers that you design at the beginning become part of your sequencing data. So these are basically synthetic sequencing information, not part of your output, not part of your sequencing, and then you really need to remove them from your sequencing. And that's in addition to the sequencing, sorry, the sequencing adapters and the primers there. So you have the adapters and the primers that are related to the sequencing platform, and then you have your probes and the primers that you need to remove from that. And those require to be trimmed up front before you do your analysis. After that, you start mapping to the reference genomes there and do the genome assembly, do the variant calling, and get the abundance and do the statistical analysis as you do normally there. As I said, there are multiple packages that can help you do all this without having to go to these various steps. And if you're doing metagenomics, there is the Kraken suite that you can use, and it will basically do all this in, I think, 2 or 3 steps for you. There's a Chime too, it's a bit outdated, but still in use. And if you're doing long read, specifically with Oxford Nanopore, they have the EB2Me labs, and they have the metagenomic approach that can help you do with this. And it's good because it gives you a little some sort of control over the databases that you can use, and you can differentiate or use different databases based on the type of host that you're working with. You can do that with Kraken too as well. So really, one thing you pay attention when you're doing your analysis is what database you're basically mapping to, and whether your target or the expected target is part of this database, or the host is part of the filter out database that you're using or not. And if you go to the Kraken 2 website, they have their own group of databases. The AB2Me as well have their own group of databases that you need to select between when you are doing this kind of analysis. If you're doing targeted sequencing, again, with Oxford Nanopore, they have another specialized platform for flu, and you can modify that for other viruses. You just need to change some of the reference files there. It's called WF-Flow, but you can basically modify it for other viruses. It uses workflow pipeline there, and you have the viral raccoon, you have the Irma and the Mira, and I believe the Mira is basically using Irma in the background. And those are targeted approach, and then you just can get your data and do the analysis directly for you, and you don't have to worry about the various steps that I mentioned up front in the analysis guidelines there. If you have a sequencer in your lab, let's say you have an Illumina, you probably have access to the Dragon tool, and they have very good actually analysis tool for pathogen detection there, and it can work with both metagenomics, and I believe they have another tool for microbiomics or the 16S. If you have the PacBio in your lab, you probably can use the smart analysis, and they have tools for that that can automate make simplify the analysis process and get you quickly to what you need. Or you can even install the Phoenix pipeline, which is a suite of things that the CDC uses, and you can install this in your lab and have this work done for you. There are, as I said, there are some commercial software like I don't know if Genius have this or not, but DNA Star and other softwares have Genome Bench, have tools for that, but those are things that you really need to pay for to be able to use them. So this is basically the standard method that we know today and are being used in for this purpose. And there are some, there has been some basically attempts or to use machine learning into this domain. And there's not a lot of work has been done as so far in this, but I think there are some promising ideas there, and probably upon their completion or more integration in this domain, then they can change completely what we do about this type of sequencing data there. And one of those, I'm gonna go through them quickly. One of them that was basically developed in 2013, and it's more about the host pathogen interaction or the protein-protein interaction. I haven't seen any much improvement in this domain, especially in the domain of pathogens, really. There are a lot of protein-protein interaction work that's been done using machine learning, but in eukaryotic systems, but I haven't seen anything, and even in bacteria, but I haven't seen anything in the virus cases, basically. The other one is the variant calling, the deep variant, and that's the tool that Google came up with or published about in around 2018. And it uses CNN networks basically. And this tool really very efficient in identifying variants. And it's a lot faster and accurate comparing to the traditional way of identifying variants, which is just mapping and calling SNPs and applying a statistical model to validate or quantify those SNPs. The deep variant basically converts the alignment into a black and white image, and then use a AI model to read these images and identify the variants where I think black is a variant, white is a background. The other one is the antimicrobial resistant prediction, and there is also the deep ARG tool that was published around the same time and use a different flavor of the neural networks there. And it's really a very important and very good one actually in terms of working with fragmenting sequencing data. If you have some sort of, fragmented sequencing data and you really want to know if there is any in the microbial sequences that you have any antimicrobial resistant genes, then this method is really very good, and you could try this as well, and hopefully there will be more improvement in this application. The other one is the taxonomic classification, and this one is called deep microbes. And similar to the deep ARG, is very good, especially when you're working with compromised sequencing data. And which is the case really in most environmental samples or pathogenic samples or clinical samples, DNA or RNA normally or usually is not ideal, and you end up with a lot of fragmentation there. And this method or this deep learning approach can help do better classification of the samples. The final one here, it's used to detection of genomic epidemiology and during outbreaks. There is not a lot of work in this domain, but it's really a method that used clustering of pathogen genomes and do some prediction of the transmission pattern there. Not really seen a lot of that, but I actually see some courses actually teaching that in some major university now. So I hope this will be something we can use in the future. I just want to say a disclaimer. I'm not really an expert in machine learning. I'm just learning my way through, and it's something I've been really very interested in getting into it. And I finally had the time to spend some time learning these approaches and how to use them in my work, as I will show you later. Now to this, basically the quick coverage of what we have, what we know, and now to talk about our own work in the lab. And as I said, our work is basically based on the idea that we're trying to basically streamline and solve some of the issues that we've seen with the other approaches that I talked about it. So we have the MIP-based approach and we have the enriched RNA metagenomic approach. I'll talk about them now. So the MIP-based, as I mentioned earlier, each MIP is designed of two legs. One is called the extension leg and the other is called the ligation leg. And after those legs, you can add all the barcoding, the UMIs, and the overhangs that you need. And those structures are connected with a backbone that basically keep them in place. And initially, this MIPS were designed to target single nucleotide polymorphism. That's where it was developed in the first place. So the area between the ligation leg and the extension leg used to be like, in the first version of this technology was about 2 or 3 bases. Now, if we want to adopt this to sequence large genomes or to basically do whole genome sequencing, we have to increase this gap or the area for the fill-in reaction. In our current design, we have this to range from 600 to about 1200. And we kind of optimize it to be around 1 kb, basically 1 kilobase between these two targets there. That's because we work with long read Oxford nanopore. Obviously, if you are working with Illumina or Element Biosciences, you would need to shorten this, and you can do it up to 200, 250, or 300 base pair. And there are some kits available for that. Once you have that design available in your hand, you can go through the approach, which basically starts by hybridization. And then after the hybridization, you go through a circularization period, which is basically do a PCR to fill in this area between the ligation and the extension leg. And then you go to enzymatic cleanup, which basically an exonuclease treatment to damage or cleave all the linear RNA in the sample, remove it, leaving you with the circular parts only, and then use PCR to amplify the target region and add the barcodes to these samples, creating this final construct that you see at the end, and that's ready for sequencing. As I said, these parameters can be adjusted according to the sequencing technology that you're interested in. Why do we use MIPS? Really, as I said, it combines both the advantages of PCR and the hybrid capture in the same one, and it's scalable and it's more specific I think it will have a much better application if we focus on viral genomes comparing to other pathogens, giving the smaller size of the viral genome. I'm not sure about some bacterial pathogens. Those are large, like in the mega base pairs. So that will end up having, probably several thousand MIPS to cover that. So I don't know about the economy of that, but for virus genome, it's very efficient to do it. So it's ideal, as I said, ideal for high throughput. We can basically combine multiple viruses in the same samples. And here's a quick comparison between the MIPS and the PCR and the hybridization capture. Keep in mind that MIPS always is a dual probes, uses both PCR in the fill-in and the probes for the hybridization. So it's both together. And so far, all aspects that we measured in our MIPS application comparing to PCR or ligation approaches, we see a really superiority of the MIPS in every one of these aspects. But the challenges with the MIPS really is there is a lot of upfront design work that you need to do to get this MIPS. And as I said, there is only one available by blind and publicly available in GitHub, and it's actually designed for targeting genic regions, only a gene or two, not the whole genome. And there are a couple commercial solutions from molecular loops and biorads, and they are mostly also for human oncology targets. And I believe, I think, I'm pretty sure Molecular Loops, and we work with them, have some kits also for viruses, whole genome sequencing, some for PacBio and some for ONT, and also some for Illumina. However, they don't really tell you any details, and you just gonna follow what they tell you to follow there. So that's the issue here. The customization barrier is something that requires a lot of work to overcome, and that's something we are trying to do here in the lab. So in our new viral MIPS design pipeline, which is a group of Python scripts to guide the design, and uses Python and uses scikit for machine learning module there. And I'll walk you through this pipeline quickly. We start by basically simulating ideal and non-ideal MIPS, tens of thousands of those. I think we have about 1 million of those designed since I simulated, based on a multiple criteria to define what will be ideal MIP and what would be non-ideal MIP, and use our machine learning tools or modules to train them on these so they can identify what's good and what's bad, what's ideal and what not. And then we end up having our basically trained ML modules there that we will use later in filtering our design. Then we start with our genome, and the numbers that I will mention here are relevant to the SARS-CoV-2. So you start first by a bunch of Python scripts using certain criteria and certain algorithmic logic there to design the MIPS, and then do some sort of local filtration based on the thermodynamics of the MIPS there, and you end up with 18 million MIPS. There is no way you can work with that. So we then use our machine learning there to filter this out, to filter them more, and we ended up having about 500,000 MIPS. That's a lot better, but still a lot. So we apply another approach to filter those out, to select basically among those 500K, try to select the best MIPS that in terms of coverage, depths, and gaps that they leave in the genome. We ended up having about 1,000 MIPS from that. That's kind of the target for us, which what we have now in design. And then we take this, we do the ONTization, basically adding all the other construct pieces, which is the adapters and overhangs and UMIs, as well as the barcodes. Once we have that, we go and do the synthesis, and for that, we use the OPOOL synthesis strategy from IDT, which is very suitable for this because it's really cost effective. And those 1,000 MIPS will cost about $400 or $500 basically to get them in a single tube and do your testing. So it's really efficient in a way where you can basically test and retest and redesign again until you basically have your final design there. And then we do the sequencing on the Oxford Nanopore test. Again, this pipeline is mainly for Oxford Nanopore and can work for PacBio as well. And if we need to do this for Illumina, we just need to change the criteria of our machine learning models there to be trained on shorter inserts and try this again. So it needs a slight modification to get it to work with Illumina for sure, but it's certainly doable there. Right now, these are the kind of MIPS that we have in the lab. And when I say 2BD, means that we are working in updating these designs, and we'll have some new designs soon, and we'll update this table. And our testing in the lab, we test them in multiple ways. We test them in single MIP panel for a single virus on sample with only known one virus in the sample, or we have mixed MIPS for different viruses on mixed samples, or mixed sample MIPS on single samples, or single MIPS on mixed samples. We have kind of variations to basically test the cross-reactivity or cross-hybridization between the different MIPS. If we have a panel of MIPS for flu, we'll mix it with a panel of MIPS for RSV or with SARS-CoV-2. Do they really affect each other? Do they work okay with each other? Do we see any dropout or whatnot? And we are testing all these different scenarios using these mixed MIPS. And so far, we are working with SARS-CoV-2, we're working with flu AAP, and we also have added H5N1 to our design, and we did some analysis on that, and with the RSV H1, RSV A and B as well in our samples. And this is the number of samples that we have been testing so far. This is the number of samples that we've been testing so far. And we have enough samples now. For sometimes we have issues of getting the right samples, but I think we have enough samples now, and we're just waiting for the new design to be updated, and we start our new second phase of testing there. So I'm going to give some examples of the results for the SARS-CoV-2 and for the influenza. In our experience, SARS-CoV-2 is the easiest to sequence, and influenza is the most difficult one to sequence, basically. Here is an example of the SARS genome and the different genes in the genome. And we have two runs, one that has the whole run produced 1 million reads, and the second run produced 4.5 million reads. And we picked two samples from these two runs, the upper one, which is barcode 8. For that, there was about 100,000 reads mapped to this sample. And the lower one, barcode 20, there's about 500,000 reads mapped to these samples. And you can see in the coverage plot there, very much the whole genome was covered in both the low and the high yield runs there, except in that area where you have this big dip in the coverage there. That's in the spike protein region, which is expected to see lower coverage there. But in the lower coverage run, we still have about 600x coverage there. I think we should, and I think we have the data for that to even try to see what happens to the coverage if we have way less than 100,000 reads per virus, basically. We use the same MIP design to basically look at different variants, to sequence different variants. The left side is the Wuhan virus, and the right side is the Omicron. And we had the Wuhan from the time of the COVID, where we were sequencing lots of the samples, and same as Omicron. And then we use the same MIPS to sequence them, and we don't really see any significant difference between the coverage for both. So the MIP panel is good for sequencing all these variants there. We didn't have any samples from the recent variants there, so we don't really know, but given how the redundant the design for MIPS, I don't expect to see any issues there. And the on-target sequencing there, you can see 99% of the sequences are on target, and only less than 1% of the sequencing really cross-hybridized to some eukaryotic targets in the background. You can see this from these numbers in this table there. So as for the CT value here, you can see the CT value versus the genome coverage. And you can see our approach with the SARS-CoV-2, we very much get very close to 100% coverage for CT value up to 29. And even after 29 up to 35, we still get very, very good coverage. I don't think it's plotted here, but we still get a very near complete sequences with this. And it's the only thing about when you're working with things like with CT value more than 30, it's variable. You don't have consistency in the results. And I assume at this level, probably the RNA is a bit more compromised, and that's what's causing this inconsistency in the results of your sequencing run. Here you can see the mean coverage depth, and as I said, we target each base of the genome to be at least covered with 20 different MIPS. So if anyone doesn't work for any reason, we still have enough to capture this particular base in the genome. And you can see this horizontal line represents about 20x threshold, and all of our coverage there, or our depth of coverage above this threshold there. That's using our current MIP design, not even the updated one. The flu genome is, as I said, is really the most difficult one to sequence, given the segmented nature of that genome, as well as the compromised RNA of the larger fragments, which is the polymerase fragments, and usually have basically truncated version of that lead to basically getting lots of reads on the ends, leaving the middle of this fragment creating this U-shaped thing. And with these dips in the middle, you really need to create a lot of coverage in order to be able to sequence this region there. So here I'm going to show a comparison between our method, the MIP, and some methods generated by New England Biolab using universal PCR primers to amplify the 8 segments of the viral genome and compare this to our sequencing and see. So here, as you can see in this pie chart, on the right side, you can see the MIP targets constitutes the majority of the sequencing run, and on the right side, you can see the NIP targets really constitute a smaller portion of the sequencing run. Here is the number, so I can make things a bit clearer. In the MIPS run, we got about 3 million read, but in the NIP run, produced about 13 million reads, almost 14 million. So the NIP approach really have much higher yield than our approach, which at the beginning, we were kind of disappointed. But when we did the analysis, we found out that ours actually mostly on target. We have 84% of our sequencing are actually on the viral genome, but on the NIPS, only 25% are actually matching the viral genome. So most of the sequencing, 75% of the sequences are off targets when you use the NIP protocol. And to compare, to put this in perspective here is the representation of read distribution per segments of the flu genome. I believe this is flu B genome. And you can see on the left side, our MIPS, most of the regions are a bit more or less similar, with the largest 3 regions, the polymerase regions, are making more than 50%, but in the NIP approach, the largest 3 regional genomes are not even making 25%. And if you compare this to the flu genome and the pieces of the pie or the slices in the middle pie chart represents the ratio of each segment to the total of the genome, basically. As you can see, our distribution or our pie is a bit similar to the reference genome. It is actually more similar to the reference genome than the NIB approach that you're seeing on the right side. That gives really us, and then I just want to point out that even with our MIPS, this run is done with a MIP mixture of probes or oligos for flu A, B, and H5N1, basically. All of them were mixed, and it's like a mixture of about 1,200 MIPS that cover all these viruses together. While in the NIP approach, you really do PCR primers, to basically 8 pairs of PCR primers to target the 8 segments there. The other part that we're working on is the enriched RNA metagenomic approach, or ERMA. And this one here starts with total RNA from the clinical samples. Just use total RNA, and we try basically to remove the host and the ribosomal RNA using different method that available in every lab, and you don't really need to buy a lot of reagents or fancy methods like CRISPR-Cas or whatnot. So our method goes like this. You start with total RNA sample. You basically do host RNA. You use basically the mRNA capture that you normally use in RNA-seq. You use it to basically capture the host RNA and remove it. And then that leaves you with the ribosomal RNA, the viral RNA, and the bacterial mRNA. Then you do ribodepletion to remove the ribosomal RNA, and we have clinical ribodepletion mixture that targets a bunch of ribosomal genes from different species. And then you end up having the viral. After that, you'll have the viral RNA plus the bacterial mRNA. And then you do poly-A tailing. You tail this to create a tail that you can use to pull down these fragments and do RT-PCR. And then you end up with enriched samples of basically the pathogens in the total RNA that you started with, and sequence this as on T. This method was still really playing with it and optimizing the different steps, but we have some preliminary results there. You can see on the right side, this is our method. And the other 2 pie charts there represents currently available methods. The middle one is direct cDNA. You just get cDNA and sequence everything. And the right side is basically ribodepletion plus cDNA sequencing. You do ribodepletion and do the middle method after that. So the only thing we can see in our method so far is it's successful in basically removing more of the host material comparing to the other methods, but it seems like we ended up removing some of the target or the viruses because we mixed those samples together artificially in the lab and trying to track the results. And I believe this is a technical glitch in our application of the protocol, and that's something we're really trying to optimize, and I have a student working on this. Hopefully this will be resolved soon. This is basically everything we work on in the lab, and these bars here show you how close to complete we are. And hopefully, once we fill all these pieces there, we will have a suite of methods, including the informatical and design approaches that can be used to do pathogen co-sequencing, and you will have everything from design tools to MIPS design to MIP protocol to enriched RNA protocol. Everything can kind of work together in one synchronized approach. The last thing that I'm gonna see very quickly, it will take me 2 minutes to mention that, is I can stop here if you want me to stop. I know I'm over time.
If anyone has any questions, maybe we stop. If not, then you can continue. Do we have
Sure.
questions? Yeah. Okay. Well, you can use the last 2 minutes then to talk about the methods to watch.
Okay, the methods to watch here is basically they use the unique molecular identification or identifiers for viral load estimation. That's a replacement of basically the current CT value. The other method is the adaptive sequencing of ONT for target enrichment. That should be a replacement basically for all the ribodepletion and host removal that we're working on. And the ML in using pathogen identification genotyping, that should improve our ability to basically identify variants and subtypes, especially with things like flu. And if are optimized and use the current state of machine learning technology there. So the UMIs is basically a bunch of sequencing, random sequencing you add to your library before any sort of PCR amplification in your library preparation. I apologize for this busy figure. I just grabbed it from the paper that talked about this in on the ONT platform. But it's really simple. As you can see, this stretch of ends that you have there, that's what UMIs are. It's just a stretch of random sequences you attach to your library prior to any amplification. And once you do the sequencing, you count the unique probes there. So it gives you direct estimate of the initial copy number of the target of interest. If you're doing this in viruses, like in our case, if we're doing this with, if we include this, in which we do include this in our MIP designs there, we just count that these unique UMIs and would tell us basically the number of initial molecules of the target viruses there. So that should be really a lot better than the CT value, because CT value is kind of indirect. It's like a cycle threshold comparing to the CT is like direct metric there. However, there is not a lot of the concept of using UMI has been really talked about, and there is a papers talking about, but it hasn't been tested in terms of correlation to pathogenicity, basically, like what you have with the CT value, where you basically define if somebody positive or negative based on the threshold or the CT value there. There need to be a lot of testing on using the MI to establish this threshold there. The other method is the Oxford nanopore adaptive sequencing, which is a method that allows you to basically test any sequence read during the sequencing itself to see if it matches a specific reference that you don't want or you want. You can do it either way. And if it matches something you don't want, it stops sequencing that and inject it out of the pore. If it's something you want, it keeps sequencing this for you. And then you can use this to remove host, or you can use this actually to select for targets of interest. basically, that you want that. And this method really now it can be used and very successful with large DNA molecules. And because the time it takes the machine to make the decision really between reading the first, I think, 150 bases and mapping them and making the decision. If your insert is short, then by the time the machine makes a decision, it's already sequenced there. But application for large genome is very successful. As you can see on the right side there, you can get really telomere to telomere genomes using this method if you have the right molecule size. And I think they are working really hard on this to optimize it. And once they solve this timing issue, it's something perhaps they will use AI on that to optimize this method there. And then this should really solve a lot of the problem that we talked about earlier today in terms of enrichment and target enrichment there. The final thing is the use of machine learning. As I said, there are already available two methods there, the deep variant for variant identification and the deep microbes for identifying microbes from compromised data. One can imagine that these two can be combined together in one approach, really to do identification, to do one machine learning approach for identification and classification of viruses and genotypes from clinical samples there. So I think I can envision how these two machine learning modules can be used in this context, but it's something really to see, and I'm pretty sure this will be done at some point and really will solve a lot of the issues that we are now facing in the sequencing of pathogenic genomics, basically. And by this, I conclude my talk, and I'm happy to take any questions.